Welcome to Let's Talk Micro. Hello, everyone. Welcome to another episode of Let's Talk Micro. I hope you had a great week. Like always, remember that Let's Talk Micro is available on Apple Podcasts, Spotify, Google Podcasts, Amazon Music, Audible, Overcast, Stitcher, Pandora, TuneIn. So, whatever you listen to your podcast, Let's Talk Micro is there. I am also on Instagram as Let's Talk Micro, no apostrophe, and on Twitter as Let's Talk Micro 1. So please go ahead and follow. On the last episode, I touched on some things that Carla Zamora went over, some types of agar or media. And if you haven't listened to the interview, which is episode 11, a conversation with Carla Zamora, please go ahead and do so. Great information. Carla is an environmental microbiologist from Querétaro, Mexico. So great information. Awesome to see what an environmental microbiologist does. So we talked about some types of agar. I went over 5 that she mentioned. 2 for gram-positive rods and 3 for gram-negative rods. More specifically for gram-positive rods, for Listeria, which was Balcam and Oxford. They contain some antibiotics and some ingredients that inhibit the growth of other organisms. This is used in the food industry for the environmental microbiologists when they get products and they want to make sure that Listeria is not in it. So it is used for the isolation of Listeria. These 2 types of agar, Balcam and Oxford, they're differential for esculin hydrolysis. So like we talked about with like McConkey, it's differential for lactose fermentation. But Balcam and Oxford are differential for esculin hydrolysis, which Listeria species they do. They hydrolyze esculin. And then we talked about 3 types of agar, 3 different kinds for gram-negative rods. EMB, eosine methylene blue, in which E. coli has a green sheen, and it's also for lactose fermentation. That one, you depending on which side of the world you're on, you might not see as much, but then hectone and XLD, those you're definitely going to see wherever you are, using in the States and in other parts of the world. With hectone and XLD, they are selected for gram-negative rods, and they are differential for lactose fermentation, which the colonies turn yellow, and for H2S production, which the colonies turn black. And for your students out there, you remember that H2S is definitely helpful for differentiate Salmonella, which is another serious organism that in the food industry you have to be aware of because it is normal flora in chickens, so you can have it in chicken, eggs, and it contaminates food products. Especially if, let's say, there's not a proper irrigation system, your produce can get contaminated with it. So you have to be careful. So this organism is H2S producing, but however, this production is not exclusive to Salmonella. So other organisms can turn black on hectone and XLD. And that's where the acronym SPACE, which is not mine, it's something I heard years ago, so I'm not claiming it is, comes in. It's a very helpful acronym to help you remember H2S or hydrogen sulfide producing organisms. You have S for Salmonella, P for Proteus, the A is for Arizona, which is a subspecies of Salmonella, C is for Citrobacter, and E is for Edwardsella. So these organisms are H2S producing. So you have to make sure that when you have H2S colonies on your agar, you correlate this by either with more testing, performing an ID, and things like that. And I also touched on the last episode about the order of streaking. This is very important. I like to repeat it a lot. But I still see techs that they don't do it. Basically, you don't want to bring any of that selective material to your other plates. So you should always streak from least selective to most selective. And why is that? Well, you can inhibit the growth on your blood plate if you're bringing materials from that McConkey, from your loop. So you always want to start with your least selective. So I gave the scenario where you have blood, McConkey, XLD, Hectone, chocolate. How will you treat those? I'm going to pause for a few seconds so you can think it over, you students out there. So we have blood, chocolate, McConkey, XLD, and Hectone. So you start with your blood, move on to your chocolate, then your McConkey, your XLD, and your Hectone. And I mentioned that McConkey, Hectone, and XLD, they're selected for gram-negative rods. So after your MAC, you can do the XLD first, the Hectone first. If you have a McConkey with sorbitol, you can vary the order of those. But the most important thing is that you cannot start with your McConkey and end with your blood. So very important, always keep the order of streaking. So what are we talking about on today's episode? Well, I have another test that Carla did mention in her interview, but I was going to cover it anyway because it's very important, which is called the TSI test, or the triple sugar iron test. In large labs in the US, in the United States, you don't really use it anymore. You probably, I don't know if there are smaller labs out there that still use it. If there are, go ahead and please, tell me on Instagram, send me a message. But I haven't seen any in the labs that I've been on. Nowadays we go more towards, like indole oxidase. We use the Vitek, the Molotov. But I know I have seen from posts from all over the world that you still use it. And you also learn about it in school. And it's still very, you get tested on it when you're taking your board exams, at least here in the US. So I'm going to go over this test today. It's a very important test used. It's primarily used to differentiate the Enterobacterialis from other gram-negative rods. Enterobacterialis, as you know, used to be the Enterobacteriaceae, a group of gram-negative rods. Most of them are intestinal flora, but some of them are not, and they're definitely very pathogenic. So what do we use this test for, other than the obvious that I said, from differentiate the Enterobacterialis from other gram-negative rods? Well, this test is used to determine whether a gram-negative rod ferments glucose and lactose or sucrose and forms hydrogen sulfide. So here comes the H2S again. So you are expecting that if you have a salmonella on this test, you should see the black, which is when the H2S is produced, black colonies. You can see black color in the agar. The colonies turn black. So let's get a little technical here. So you know that when I'm saying this, I'm talking about my reference book, Bailey and Scott's Diagnostic Microbiology, an excellent source, excellent reference book. So this is a tube where the agar is slanted. And what's in it? Well, you have 10 parts lactose, 10 sucrose, 1 part glucose, and 1 part peptone. It also has phenol red and ferrous sulfate as indicators of acidification and hydrogen sulfide production, respectively. Maybe you might not be familiar with the ferrous sulfate, but definitely with the phenol red, you hear that a lot, right? So it's an indicator of acid production. So when it will change the medium to yellow when acid is produced and the pH is lowered. So this agar, it's basically composed of 2 parts. All right, so we talked about the ingredients that it has. So it has 2 parts. There's the slant, which is obviously it's slanted, it's tilted. And then you have the bottom, which is called the butt. butt. So an organism that ferments glucose, it will turn the butt yellow. And an organism that ferments glucose, lactose, and sucrose turns the butt and the slant yellow. So why is this color change? So the agar it's like a red color. So why does it change to yellow? And that's what I talked about before, that when acid is produced due to fermentation, the pH lowers and then the color changes from red to yellow. And then an organism that produces hydrogen sulfide turns the agar black. So once again, I talked about the space organisms. So salmonella can turn it black. So before we move on, go ahead and once again think about H2S producing organisms. Space: Salmonella, Proteus, Citrobacter, Edwardsella. You don't see Edwardsella as much, at least in my side of the world, but you definitely deal a lot with Proteus and Citrobacter, and we'll talk more about Proteus in another episode. This is one of the greatest things about this field. There's so much to talk about, so many little components. So today we'll just touch on the TSI and the reactions. Also, if gas is produced, this is noted by the lifting or breaking of the agar. So how do we inoculate it? So very simple, there's a tube. Like I said, it has a slanted portion, it has the bottom portion. So there's gonna be a little bit of streaking, but none of the quantitative or 4-quadrant streaking. So you do the stab and streak method. So what's the stab and streak method? So you grab your colony, you stab the butt, and then as you pull back your loop, then you start streaking your slant. And very simple. So it's just a stab and streak. And then you incubate it, and then you read it at 24 hours. So how do we report it? We use the terms A for acid and K for alkaline. So we read it slant over butt. So if the slant and or butt are yellow or acid, we're going to use the letter A. If they are red or alkaline, which is, remember, fermentation lowers the pH, causing acidification, and there's a color change, we call it K. So these are the two terms that we use, A and K. So if you have an alkaline slant and no change on the butt, so basically the whole thing stay red the way it was. So in this case will be KK, K over K. And what does this mean? Well, it means that the organism does not utilize glucose, sucrose, or lactose, like it doesn't ferment it. So then what if we have an alkaline slant and an acid butt? So it's K over A. So what does this mean? The organism ferments glucose only. And if we have an acid slant and an acid butt, AA, this means that the organism ferments glucose, lactose, and or sucrose. If we have a black precipitate on the butt, that means that hydrogen sulfide has been produced by the organism. And then if we have gas, bubbles, or cracks, which indicate the production of CO2, carbon dioxide, or hydrogen. So these are a lot of terms, especially, if you're a tech, you're more used to this. But if you're a student and you're trying to learn this, let's go ahead and go back real quick. That way we can reinforce this. So for reporting, we use the terms A for acid and K for alkaline. We read it slant over butt. So you can have two different colors. The agar is red and then it changes to yellow. So if the color is yellow, you report it as A. If it's alkaline, red, no color change, you report it as K. So if you have an alkaline slant and no change on the butt, so meaning that everything is red, alkaline is red, the agar is red, so there was no change. So that'll be K over K. So the organism does not ferment glucose, sucrose, and lactose. If you have an alkaline slant and acid butt, K over A, the organism ferments glucose only. And then if you have an acid slant and an acid butt, so the whole thing is yellow, it'll be A over A. And this means that the organism ferments glucose, lactose, and sucrose. If the butt is black, if there's a black precipitate on the butt, that means that hydrogen sulfide has been produced. And then if you have bubbles or cracks in the agar, this indicates that gas has been produced, that CO2 or hydrogen has been produced. And that's how we read it. So let's go ahead and start talking about some results, right? Let's talk about our E. coli. So E. coli can vary with the results. You can have K over A over A, or K over A with gas. So we can have alkaline over acid, acid, or alkaline and acid with gas. What about Klebsiella pneumoniae? A over A with gas. So acid slant, acid butt with gas. What about our friend, quote unquote, Salmonella? So K over A. So alkaline slant over acid butt with gas and H2S. And then we have Pseudomonas aeruginosa, which is K over K. So now if you're thinking about this, you're going, whoa, wait a minute. He said this test is used to differentiate Enterobacterialis from other gram-negative rods. And then he's been talking about Enterobacterialis. He gave me the reactions for E. coli, Klebsiella, Salmonella, and now he's talking about Pseudomonas aeruginosa. And why is that? Well, it's very easy. Now let's see if you have, from what I've been talking about reactions, on my episodes. Let's think about this. Is Pseudomonas aeruginosa an Enterobacterialis? If you're thinking yes, then go ahead and study a little bit more. If you're thinking no, you are correct. And why is that? Well, if I don't know anything else about Pseudomonas aeruginosa, I know one thing. It is oxidase positive. And like I talked about on my episode about the oxidase, on the oxidase episode, if a gram-negative rod is oxidase positive, it is not an Enterobacterialis, because all Enterobacterialis or Enterobacteriaceae are oxidase negative. That's part of their trait. That's the way they are. So then why are we going to use it in this? Well, it's definitely a control, right? We know that Pseudomonas is not a fermenter. It's a non-fermenting gram-negative rod. So if you put it there, it's your negative control. You're making sure that the actual media works, placing an organism that you don't get a false positive. So you're expecting Pseudomonas to be negative. You inoculate it, and then you see the red color. So you know that you don't have a false positive result because it is supposed to be negative. So it's a great negative control. But remember, and I want to reinforce this again since I said it, and I talked about it on the oxidase episode, Enterobacterialis are oxidase negative. So if you have an oxidase positive organism, it is not an Enterobacterialis. So definitely keep that in mind. And I mentioned that you incubate it and read it 24 hours. You can read it from 18 to 24 hours. But you make sure that you shouldn't read it beyond 24 hours. And this is because, there can be an oxidation of the fermentation of the lactose and glucose. enter sucrose, and then the slant can go back to the red color. So there was a fermentation, and then it incubated longer, oxidation took place, and then the color reverted again to red, giving you a false negative result. So make sure that when you incubate it, you read it from 18 to 24 hours. And normally most of this test, that's the time that you should read it. That's where the organism is at its best. So keep that in mind. If you're not gonna be able to read it after the 24 hours, make sure that you don't perform it or just let someone know so they can do the proper reading. So I gave you reactions for Salmonella, for E. coli, for Klebsiella. So now, knowing what you know, if I give you a Proteus mirabilis, what would you expect the reaction to be? Well, think about it. Proteus is a non-lactose fermenter. So now from what you've seen, you can expect the slant portion to be alkaline, K. It is a glucose fermenter, so you expect that to be A. And then from your favorite acronym SPACE, it's an H2S producer, so you can expect H2S production. So it will be alkaline over acid with H2S production, or K over A with H2S. What about Shigella? Well, the same thing. Shigella is not a lactose fermenter. It does ferment glucose. Shigella doesn't produce H2S, so you're not expecting that. So it will be K over A, alkaline over acid. So that's one way to think about it. So the glucose fermenters, when the organism ferments the glucose, it happens on the butt. And then when they ferment lactose and or sucrose, it's on the slant. So those organisms that you put in McConkey that are non-lactose fermenters, they are expected to have an alkaline or a K reaction on the slant. So very simple. So keep that in mind. And today if you learn about the stab and streak method. This is something that you heard about, probably learned in school. If not, if this is the first time you hear it, it's very simple. So you stab the butt, and then as you're pulling out your loop, you start streaking your slant. Very simple procedure. So remember that. Remember the acronym SPACE. And like I mentioned before, maybe this test, you might not do it in your lab here in the United States. In other parts of the world, it gets done. But this is something that you will definitely get asked about. For example, here in the US, there's an organization called the American Society for Clinical Pathology, or ASCP. They have no relationship with this podcast. And they give you a license, so you have to take an exam with them. And then when you pass this exam, you get certified by them. And the ASCP, you can basically work in any state here in the US. And this is definitely one of the questions that they ask. So it's very important to know this test, know how it works, and know their reactions, because even if you might not see it at your workspace, you will definitely be tested on. So there you have it. So I just want to touch a little bit on Pseudomonas aeruginosa in case I confuse anyone. So it is used as a control in the TSI agar because it is negative. It is a non-fermenter, but it is not part of the Enterobacterialis, as your students should know. Just on the oxidase alone, it's not an Enterobacterialis because it's oxidase positive. And that, my dear audience, is the end of this episode. I hope you enjoyed listening about triple sugar iron test, because I did enjoy talking to you about it. Continue that motivation. Continue bringing that passion into what you do. Thank you for all you do. Continue studying hard if you're studying. It's always great when you are very passionate about what you do. So definitely continue bringing that. Stay safe and continue talking micro. Once again, it's been a pleasure. I hope you have a great week. And until next time.